First of all, thank you so much for the invitation. I know it's a crowd with a lot of spatial omics enthusiasts, and really happy to share what we've been up to with testing out different spatial omics and building a large community resources as a group. Um, and like everyone else in the audience, we're very interested in how to map, understanding how to map comprehensively across multiple scales of biological systems. Of course, we cover a wide variety of ranges of genotype to phenotype, where it comes from molecule to cell to tissue to organ to the system. And of course, spatial biology really gets you to map molecules in situ, essentially. Back when we had bulk RNA sequencing, we basically had one data point per group. Now with single cell, we get to make a granular detail level of resolution by having a single, like by understanding counts per cell, which we can color each of the cells by its identities and gene expressions. But now we can also reorganize these dots and put it into more of a spatial context, not only the counts per cell, but also the location as well. And as these technologies develop, over time we saw a lot of large projects that are popping up from human genome projects where it really understood the genome sequences, all the mutations and biological outcomes of these. And Human Cell Atlas was also a large initiative that really understood gene expressions, identities, and how the cell types are located, organized in the body. What was missing now that spatial omics really enabled us to do is to have a more context, spatial locations of the transcripts, how they're organized and how they are interacting each other in the physical coordinates. And of course, the beauty of this is being able to layer all of these different modalities and information on top of each other. And of course, when the new technology comes along, we always have those questions of like, do we have enough data? Like, how do we build a reference? How do we know it's a standardized? Um, what are the methods to computationally model and extract the most out of these assays? And of course, along the way, what are the methods, tools, and what kind of insights we can get, given that we can generate a quality data. And over the years, we know that there are so many different technologies and so many different flavors of technology comes out. Um, we have virtually tested most of the platforms that are out in the market. And when a lot of people ask us, so what's your favorite? We always tell people that it really depends on the experiment because I think each of these modalities will come with its own pros and cons and one's strengths and niches. But it's been really exciting to see how all of these different chemistries and different detection methods and different pro panels have been really putting together under the same goal, really, to understand a better biology. And of course, when the first round of technologies came out, we put together a review paper. Probably it's outdated at this point, but we do put together reviews and our outlooks of it once every few years. But this is really what really got me excited about the spatial technology more than the coolness of the of the assay. 19th century or decades ago, we've been stuck to histopathology, where once there's a cancer or any kind of disease, diagnostics happen, pathologists will look at the H&E or maximum ISC assays to look at how the tumors looks like, what kind of staging or grading information you can get out of these, and basically decided whether these patients has tumor or not, whether this patient needs to be treated or not. Now with the molecular pathology, we get to have a lot of companion and diagnosis with next generation sequencing, PCR, FISH assays to better stratify and understand what groups of patients or what mutation subtypes these patients do have in their tumors, which will lead to a better response and worse response in the therapies or prognosis as well. Now, I think with the spatial multiomics, we get to understand these like two different layers or two different types of information on top of each other from the same exact sample. And now we can locate where these molecular changes are happening on the histopathology slides or on the actual locations of the tumor. Now we're trying to, I think now with those information, we can finally get to the personalized diagnostics or personalized assays as we develop this further. And of course, we need more data to get there. And that's why we started this large initiative, what we call a Spatial Atlas of Human Anatomy or SAHA project, where we wanted to build a reference data map from healthy adults from wide ranges of age, gender, and ancestry, and take 30 or so normal organs and run spatial omics assays to understand all of these cool informations that we can get from the spatial omics, from spatial expression, tissue architectures, to how the cells are co-localized, forming networks, what kind of interactions are happening, and if we can also integrate and the multimodal layers so that we can do a full integration of the biomolecules. And of course, the huge driving force behind this is we need to really understand what the normal is to better understand all the diseases and all the interesting biological questions that we're asking. And through the years, I think this was the biggest takeaway from our work, right? We were able to build the interdisciplinary cross-organization collaborations. And it's been amazing to realize how many expertise we need now that we are merging these like pathology and clinical research into more of a molecular biology and genomics computational research. So we do have a great team of pathologists, clinical researchers, core directors, computational biologists, to also like industry partners and other academic partners as well, who's running spatial omics centers, who's developing platforms, who's been developing foundation models or running clinical trials, up to places like NVIDIA or Bruker, where they're actually building solutions, scalable solutions for other customers as well, how to build a better, more scalable technology, but also the computational platform as well. We all came to a point where we really believe in generalized connected model is needed, and we need to standardize these workflow for that can work for a lot of wide range of the organs so that we can scale this up down the line. A few years into the projects, we recently got our preprint out. Currently, the paper's in revision, where we were able to profile a little more than 100 patients of worth of the materials across the GI organ and immune organ systems. Of course, a lot of these samples do have a disease pair, so that we can start asking those interesting biological questions that can only be asked in spatial. We also by default had a lot of cosmics and RNA protein, but also other spatial modalities as well, sometimes single nuclear RNA sequencing on these ribbons to build as comprehensively as possible. And as we are speaking, we are also generating more data sets down the line, which I'm going to briefly mention, where our second phase will be more on the genitourinary system, which already started with the prostate, but we do have other organs that are in the queue, but also in generation right now. So I tell people that we are building Google Maps for all the cells, in a way that we want to now not just looking at what kind of buildings are there, what kind of cells and neighbors are there, but we are actually building more comprehensive map of like how the tissues are organized, as if you're seeing the H&E into a real in the real like physical space with all the molecular information on top. And a lot of our data sets do use Cosmics, but this process pretty much applies to any other spatial omics platforms. Regardless of where we start, we always look at the H&E, make sure they're structurally well maintained, they're in good quality. All the biologically interesting regions and typical regions are included in these samples. And for Cosmics specifically, we do have to choose specific field of use, which we'll call as FOV, and use the morphology marker kits to identify the structures of the tissue. And based on these markers, we get to segment the cells, or like, draw the boundaries of the cells to understand and build a mask. And based on the RNA or protein transcripts that we can capture from these platforms, we get to map it back to the segmented mask that we have created. And at the end of it, once we map those back, it becomes a cell by G matrix where you can cluster and understand niches, understand cell types, expression, and all of these questions that we've been asking in single cell, plus all the subcellular resolution questions, as well as the physical location information as well. But I think the true beauty of this atlas is two things. One, we are able to go back and forth the scales from the histology images that we can see on top of this table that we typically see in the clinics, honestly, and go all the way down to UMAPs where the researchers do see every day, and going back and forth different scales and different resolutions to ask different types of biological questions along with these expertise. And then the other thing is because we are using imaging-based platforms, we do, I mean, yes, we do have a different types of biases that is created by the set amount of a panel, but relatively it was a lot more straightforward for us to integrate these data sets and put all of these different organs into on one reduced dimensional space and start asking how these organs, how the structures do get conserved or similar or different from each other. And one very interesting thing, and of course, we started a lot with the AI approaches, and of course, we can start with the histopathology. Another really interesting moment is that we've been realizing, and we've been in awe of like how much of the information that histopathology images already have contained in these images. We first took a look at the H&Es and patched it up and ran through the multimodal foundation models, and we can already, even without the molecular information, we can get broad ranges of the broad domains or tissue regions that can be captured from just from these imaging layers of information. And of course, we can use this to not only compare with the pathologist annotations and our knowledge about what these regions are, which we can now unsupervisedly, in an unsupervised manner, look at how these different regions are captured in our samples, but also going back to our molecular level informations and how much of a proportion, how many cells are actually found in these specific tissue domains and regions that we can capture from H&E. And even when we get into diseases, we get to understand just from these images what are the pathways or what are the text labels that are popping out in these IBD samples, for example, over here relative to healthy ileum sample. And a lot of these pathways, a lot of these observations do correspond to the molecular level pathway analysis when we take all of these cells in inflamed regions and look at what's the top upregulated pathways. And we can go a little more further and look at how we can look into gene expression level information by simply looking at the similarities of the text. We probably are not perfect in this, but still able to identify the clusters of the genes that are very similar in function for each of the histological regions. And of course, there's a lot of different models that we have in development that to bridge this gap, basically going just from H&E, can we start training all of these molecular information together and fine tune them to do a better annotation or do a better prediction of these gene expression patterns? And can we really make a generative model for people to use it for clinical applications, which I'm going to talk towards the end of this presentation. And this is a couple of examples what we're seeing already, right? If we do I mean, I mentioned we can do a lot of domain detections just from H&E. If we actually take a look at the molecular layer of information, as you can see on the left, a lot of the lymphoid structures, a lot of immune signals do get lost as we have to increase the patch size window. And of course, when we start to ask really granular questions of what kind of immune cells, what subtypes are determining the phenotypes of the tissue, we need to consider how these gene expression fluctuations happen, what kind of cell proportions or cell identities in that specific regions can be very important. And this is something that why this really shows why molecular maps are still very interesting and important to profile and what we are trying to fine tune with these existing models to bridge that gap. And as we do those, I think a lot of the conventions or a lot of the thoughts are now beyond just taking a single like cell by gene matrix, and we started thinking everything as more of a graph structures and networks. And every time we visualize, I think now we get to question how these are all individual cells are all connected, what kind of new cells are being connected, and how has been one of our questions. And of course, another moment that we had with these data sets is it's no longer an interactions of the cells by like one cell by one cell. We get to really define the motif of the cell-cell interactions and start looking into how we can go beyond just looking at cell A is interacting with cell B, but having multiple cells interacting through multiple means of a receptor ligand as a features, and where does it gets where these motifs are getting enriched in specific regions. Here we're showing as an IBD, but I think there's going to be a lot more interesting question as we think about cell-cell interactions as the groups of cells interacting in multiple directions. And we start to build how these interactions and graph structures gets generalized across the GI tract, for example, over here. And of course, we do have a heavy data sets on GI and immune. I think it can go further if we start expanding into multiple different organ systems. It could be, and very excited to look at how these structures that we have found and how the representative cell interactions or cell network shapes that we can capture from these GI regions do get conserved and lost or change over, for example, over here, how they change over the GI tract, go through the GI tract, but also how these immune structures vary across multiple organs. It's something that we're looking into right now. And of course, the protein has been really interesting as layers of information that we've been using mainly for validation, as well as better annotations of the cells and identifying substates of the cells. For example, all the receptor ligand interactions pairs that we've looked at for that are spatially resolved can be validated with the protein layer by looking at how they are colocalized with the cell types that we found, but also where the protein is expressed and how much of a colocalization of the protein signals do we see in this tissue. And of course, there's a million things that we can ask. And because we are still a limited group of researchers and we believe in open science, that once we release these data sets, everyone can start asking all of their own questions, all of their own expertise questions with this data set. So we are, of course, sharing a lot of the code base and data with Zenodo and GitHub as an open source, but also we recently been putting everything on the AWS Open Data program as well. But also building interactive browser for people to look into and start exploring without having to code these. This is one example where we really focus in collaboration with Aspect Analytics, look at how we can align the H&E with all of these molecular data sets. But also with Biotree, we've been building more of an interactive database and Explorer where you can access and start playing around, get all the data downloaded, only the regions that you want, except for whereas for Zenodo, it's going to be pretty much downloading all integrated objects. So you can pretty much do, or I think it's more of a limitations of what kind of questions you have and start looking into all the genes that's been of your question, where it's expressing how much it's expressed, what kind of cells are there. But of course, as we're building a large set of data sets, we want to make sure we standardize everything. And it's still despite the amount of data sets that we have, we still are rigorously working for how we can improve the quality of the data sets. For SAHA, specifically the data set that we are releasing, we've done a lot of QCs around these 3 different criteria of course, what can happen during the sample collection time and what in the pathology core, how we can process them better, but also what are the tissue-specific quality measurements that we do have to do. A lot of the times we screen many of our tissues using RNA-scope and by pathologist evaluation. And we've seen a lot of correlations with how much RNA is captured or how much RNA is in there that will directly correlate with the quality of or amount of information that we have from that given tissue. And a lot of these questions also led to a separate project in collaboration with Dr. Jasmine Plummer and Luciana Morletto, where we took some of the SAHA samples as well as other cancer samples from other institutions and ran consecutive sections across multiple modalities, but also multiple centers so that we can question if we process everything exactly the same way, if we have exactly the same slide or write consecutive slides that were sectioned at the same time and ran in a different machine at different institution, how much of a variations are we seeing, what kind of quality metrics that we do have to consider to say this is a successful run. And through the process, the papers available from Nature Biotechnology, but also I think the tools, and we do have a protocol IO page for all out of the standard operation procedures for people to look at what we've done across all of these three different centers separately, but also had the tools of that we can automate the calculation of quality metrics and scores for the users who are generating the data, but also look into how where your own data sits compared to other data sets that's in the public or the data sets that we've generated so far. So we're currently at the stage of scaling really this as a Sahab for a global consortium. It was initially a project that we started from Cornell. Rapidly had so many different industry partners and other academic collaborators who's been developing or pushing the fronts in different ways. We're officially starting as like more of a global consortium where with all of these like data set that we are generating from Saha, but also from other initiatives, but also the technology that we are developing as a standard operating procedures, but also newer technology implementations that we're doing, but also through the process, really good collaborations, collaborators, and industry partners that that are all working together along the same goals to get the maximum out of all of these spatial data sets and try to understand better on how these tissue architectures are leading to the phenotype. And these are five different areas that we're pushing forward, of course, getting more data sets across additional tissue types, especially we're going into the genitourinary organs, including bladder, kidney, and reproductive systems of a male and female. And of course, along the way, we do get a lot of bladder cancer, some of the really developmentally interesting questions like ovotestes or other specific developmental defects of some of the organs that we have in the pathology clinics. And of course, developing specific methods for these organs and implementing higher plex platform for us to better profile the biology and being able to see where we can develop new models and computational methods to better translate this into a real actionable items. And as we were building this forward for the past 4 or 5 years, I think there has been a huge leap in the field of different aspects. I think every year when we go into different conferences and when we talk to different spatial omics partners, I think these are 4 different fronts that those platforms have been developing into. Of course, the throughput where we get to profile more cells or a larger area given the same price or sometimes at a lower price. A lot of the platforms are going for multi-omics and of course higher resolution of the technology. Everything's now subcellular, really trying to get those information out of the cell, but also building a good framework for everyone to democratize the data, but help disseminate it better, but also to extract the most out of these complex assays. And for the rest of the time, I'll try to explore a few different directions that we're pushing to. One is going 3D. Of course, now apart from doing the 3D reconstructions of the consecutive sections, we can now also do the tissue clearing and do the 3D spatial transcriptomics. And this is one example where we get to profile pretty much the entire eyeball of the mouse retina at the subcellular level. And this is an example of the Stellar Omics platform where we can do 3D spatial transcriptomics. And these are actually the retinal samples that has been housed in the International Space Station for 6 months. And we get to really see the impact or molecular level impact of these, like what kind of stress and radiation, microgravity specific effects that these retina had to go through while they're in space. And of course, as we go on 3D, it's just more than just simply layering these dots in the z-axis, but also it requires 3D segmentation, and it has now like a new sets of challenges. Now we have to add it now that we're adding a new dimension. Of course, there are tools that are available like MetSem from NVIDIA that can be used to do the 3D segmentation. It's still not perfect. The neuron layers and still needs to be worked out. Better segmentation optimization needs to be done. But still, it's been very mind-blowing to see how much of a signals that we've been losing by taking a single 2D sections and capturing those. So the Z-stack windows that I've been showing is basically the typical 10 micron sections that we can show in spatial omics. And by having the 3D spatial, we get to really not only get the full transcript information of the full cells for a lot more, but also be able to capture signals in a whole different depth. So we're still at the stage of really exploring the data to see what kind of specific, where is the layer that has the most impact, what kind of gene expression profiles and changes that are found in spaceflight. A lot of these panels are already available right in our panels, but we also are questioning a lot of these based on the other single cell and geomics spatial resolved pseudobulk signals or gene signatures that we already have. And hopefully in a few months or so, you guys can see through the publications or through other outlets what kind of findings that we do have related to spaceflight or spaceflight associated neurokiller syndrome related results from this research model. And talking about getting to adding another dimension, time has been also one interesting dimensions that a lot of people talk about. And of course, we do now have technologies for running spatial multiomics experiments in vitro and start questioning how we can incorporate time into the spatial omics assay. Specifically using Selenium cell cage enclosures, we get to now map all of these protein level informations and cell morphology informations with the mRNA profiles of the cells that we enclose in the cage. Here we're specifically interested in understanding how the senescent cells are showing how the morphologic changes and phenotypic changes of the senescent cells in cancer is specifically affecting the phenotypes. That's why we are mapping those back into mRNA profiles. But also, apart from like interesting biological questions that we can ask, we can also practically do the lifestyle captures and do a large scale screening for the CAR T cells to find out what combinations of like E to T ratios or what kind of the clones of the T cells are most efficient in killing the tumor and being able to use this more for translational purposes as well. And lastly, as we gain different methods that can increase the throughputs better, we are also actively adapting the STAMP to pretty much transfer ourselves from thinking single cell to like think about spatial single cell. As we do have a higher resolution than just simply running single cell. We get to understand how the subcellular resolution of the transcripts looks like, but also being able to profile a lot more cells in a single slide or in a single library by putting the cells onto the tissue onto the slide instead of doing the isolations and running the single cell library. And of course, we get to assess this technology when like right before it came out and ran a large AstroStem study across all of the astronaut PBMC samples that we do have in the lab and we're able to really load like 25 more times of the cells per sample after optimization and being able to basically substitute the typical PBMC profiling that we do and single cell multiome profiling we do and substitute with the spatial. And the ease of this, the one other benefit of the system is we get to fix them right away without having to preserve it fresh and having to process them extensively before we ship them back to our lab. And when we tested out for the cells that we have used already or have already generated single cell data sets for, we were able to basically from like a couple of slides, we were able to get more than 5 million cells, or 3 slides, we get more than 5 million cells, and we were able to load 40 times more cells compared to what we used to do when we take 10,000 cells out and preparing library and developing and sequencing for single cell. And of course, the assay quality or data quality is pretty nice. I think overall there are still segmentation errors, or there are still like noises that we can capture across these UMFs when we project it onto existing cell profiles. But we get to also identify some of the really rare cell types a lot more in proportion than when we normally run single cell, purely because, well, two, like one, because we do capture more cells in general, but two, because we fix them right away without having the cells to go through extensive prep procedures. And this is one example where we can really start to question like what kind of cells or what kind of fluctuations do happen in one of the crew members over time when they had like a couple of different space flights and multiple missions. Um, but also going all the way down into the 3D again to look into now just not looking at the expressions and what proportions of the cells are there, but also what are the colocalizations or what kind of informations are captured in subcellular level, and specifically using pixelgen of the same astronaut samples and look into what are the polarity of specific immune molecules that we can capture from these cells. And of course, start looking at it more systematically. You look at what's exactly changing or what kind of subcellular features are changing during or post-flight relative to pre-flight, and being able to quantify the landscape of the protein colocalizations or protein polarizations across with the space flight. And lastly, I think the huge excitement in terms of like actually bringing all these fun technologies into the clinic. We've been thinking that same sample multiomics has been a lot more useful than like going 3D, which takes a lot of tissue clearing process, or like adding temporal elements needs a lot more in vitro or a lot more of a experimental setting. Whereas for clinical cohorts, I think what really matters is to how to find clinical markers and low-plex panels that we can run across large scale cohorts to understand what are the how to generalize all of these architectural differences, how to generalize all of these spatially related findings of these datasets that we generate. And of course, Singular G4X platform has been one of the good solutions as we offers low plex but high throughput options that can layer all of these H&E, proteomics, and transcriptomics, and now direct sequencing as well. So we were very interested in asking the questions of like where this can fall into. And of course, in the tumor biology, we care a lot about the screening of like what are the factors that will impact the patients to develop a tumor or not, that has more propensity to develop a tumor or not. If the patient gets a tumor, um, how do we diagnose these better? What kind of staging and what kind of molecular subtypes that we can test for these patients to have a better, more efficient and timely treatments? So spatial omics has been used, and also a lot of the molecular pathology has been a lot used in this specific goals, right? Look into what are the subtypes and how to better stratify the patients. What we're very interested in is in the third part, where it becomes more of a surveillance, and having seen a lot of the patients in the clinics, but also having seen like friends or family going through this process. Even after getting the surgery, patients do have to come back for CT scans and blood works to make sure the metastasis or recurrence doesn't happen. And it becomes more of like a repeating cycle until if the patient gets negative, you get retested in a few months, few years. And once there is a recurrence or metastasis, you go back into all of these complicated system where you have to go through the diagnosis and everything again. And there are a lot of questions and methods that people do develop to better predict the metastasis. Um, Breast cancer, you do have an oncotype. Colon cancer, you don't really have an equivalent molecular testing. A lot of the people use Immunoscore or TMM staging for prediction. It's still not perfect, and it cannot really predict where the metastasis happened. And it cannot really predict the individual level biology as well. So, as the tumor, I mean, we know the colorectal cancer goes into a lot of the cases in liver, some rare in peritoneum, and some in lung, rarely in the bone or brain, but it does happen. we wanted to see why or how these colorectal cancers travel to or metastasize into these adjacent and like far distant organs and what kind of molecular signatures we can find. So far, the genotypes or molecular signatures haven't really worked. Whereas we do know that there are multiple cell types that are driving, and there are multiple hypotheses already that already exist that may contribute to the propensity to go into liver or propensity to go into lung as opposed to liver. And a lot of I mean, the huge gist over here is we now understand that it's not just the tumor cells or the mutation those tumor cells have, but it's also the fibroblasts and immune cells that are interacting with these tumor cells that are or that are pushing these cells into specific regions. And also spatially, we also know that these organization changes can capture these biological changes, right? Here we're showing as an example of a colorectal cancer patients where we show from all normal muscles all the way on the left side. And as we go through the regions where there's metastasis happening, and on the rightmost side, you can see the metastatic cells, and you barely see the normal muscle cells, and you can even see the ductular structures of the colorectal cancer cells in these regions. And we get to basically marry these two together, right? From the H&E images that we just saw, where we can see, hey, here's a tumor, here's a stroma, here's the normal cells, which is basically like a black and white images, to a colored image where we can understand these are pre-metastatic niches. These are the cells or tissues that likely will go into specific that has a specific immunosuppressive microenvironments or that specifically can respond to therapies. And we get to really understand these molecular level information and identities of the cells in a much more deeper sense, eventually can help the patients to better, or clinicians to better understand about these diseases and diagnostics as well. And from spatial omics data sets, we already know that there is a huge interpatient heterogeneity. Not only the heterogeneity itself, but when you look at these crypt regions and take a couple look, we know depending on how deep the tissue section is, it's different types of enterocytes or different types of cells that are filling up the regions. But also within the tumor, we get to, even if we see the tumor cells over here, it's actually composed of very different quote unquote immune microenvironment that they're exposed to. Here we look simply looking at the lymphocyte tumor ratio over here, but sometimes we do see a different tumor subtypes within these regions as well, all from the same patient. And of course, looking at this intra-patients, we started asking, like, if you're seeing this much heterogeneity, like what are the intra-patient variability would look like? And also started like trying to find a way to generate a large cohort of datasets or atlases to understand this better. So as I mentioned, for same sample multiomics, we try to use Singular G4X platform to generate a large retrospective study with extensive clinical metadata, including the mutation information, the genome stability, different staging information, metastasis records, organoids, and different other in vitro models available, and try to build a large, comprehensive spatial resource for us to really understand better of where the metastasis happens, but also for the community to ask all of these questions. And we call this project SpotMed, where we really want to understand the spatial predictors of tropism and metastasis, mainly what makes the cells to go into the liver when they metastasize, and can we predict them? And we're able to generate a layered information of H&E, protein, and RNA, all in the same sample of typically 1 by 1 centimeter square per sample. So we specifically built the panels for these on top of the 280 colon cancer panel that I already have. We built intent microbial probes that are known to be related to the colon cancer, mainly Fusobacterium. 62 custom genes that are derived from the matched bulk RNA sequencing datasets related to metastasis. Other known therapy targets, literature related to organotropisms, and also 12 different targets derived from the spatial omics assay from the colorectal cancers dataset that we had. And based on those, we already started from the patient metadata information to make sure our cohort is well balanced and covered. We do have a really good mix of patient samples, and this was in collaboration with Yonsei University Hospital in South Korea. And that recapitulates the known CMS subtypes of the colorectal cancers, and there has been a wide range of metastasis and outcome information. And one beauty of this is a lot of these data sets, we do have a matched bulk RNA sequencing for any kind of mutations or any like bulk level signal matching of these spatial omics data. And although we recently started this project, and this is some of the snapshots of the data set that we have generated so far. So far, we've done around 300 samples so far. And as we're speaking, we're actively sectioning and generating more data. But even with 350 plex panel or around 350 plex features, we get to understand most of the subtypes of the colorectal cancers and all the immune infiltration, specific immune cell reservoirs, how they're structurally resolved, all nicely can be recapitulated just from these data sets. And it's a matter of us starting to ask all of these questions and how we can generalize these across. And of course, as I mentioned, we've built in microbial probes to understand how the biofilm gets formed and what kind of immune responses are associated with this in their cancer prognosis, but also have smaller cohorts of patients that have responded and not responded to immunotherapy to identify like immune activation spots, but also look at their spatial heterogeneity when it comes to immunotherapy response. And we're just tapping into the surface and still actively generating and data and understanding what we can do with these. But we're already seeing what's conventionally known about like macrophage polarities and T cell infiltrations of some of these responders and non-responders of the patient cohorts, and start asking all of these how we can basically have some level of spatial predictor of the from these data sets to predict how they're responding to therapy, but also what kind of risk that they're carrying just by taking these samples and biopsies from these primary tumor. Of course, as we build a large set of data sets, I started questioning like how much data set do we really need? And is 1,000 enough given like a lot of genomic studies has been putting together a larger sets of samples to ask a stratification problem or like asking the questions of how to generalize the patient information. The amount of information that we're getting from the 1,000 patient alone is very equivalent to the previous like really large scale, like 100K genomes projects, because we are getting so many subcellular features and number of cells from each of the patients. And eventually with these large data sets, we would love to basically streamline all of these fragmented diagnostic procedures where we do have an H&E from pathology lab and pathologists, separate layers of information of the next generation sequencing from a different sets of samples that were collected, not the exact same consecutive section that they have for where they diagnose a tumor. All of these different blood works or different diagnostics and assays that we do have to run in different samples. If we can reduce this information and start getting some molecular information by generating a large data sets, training to reduce to a minimum feature of an assay that we can run on the patients, and being able to diagnose most of the information from the whole slide imaging from the pathology. So that's where we're going. And of course, to get there, we do need a lot more data, a lot better models and diagnostics, as I spoke in the beginning of the talk. But I think with all of these tools that are being available, all of these different like agentic pipelines that we're building, and also a great amount of collaborators and data sets that we're building, hopefully we can get there faster. And of course, to get there, we do start to have a different set of challenges, right? When we started generating these colorectal cancer data sets, I started asking around like, hey, have you ever built a UMAP out of the billion cells? Or like, have you really used all of these both protein and RNA informations properly, other than just trying to integrate or trying to annotate it back and forth? How can we really connect these RNA and protein and histology scales all together. And these kind of different questions and different sets of challenges will pop up as we build this together. But I believe in a team science that will get there and a lot faster than what we used to. With that, I'd like to thank all of the collaborators and people who's been really crucial to a lot of these different parts of the projects, but also more importantly, all the patients and their families for supporting and providing samples and making all of these research possible. Thank you so much for listening and happy to answer any question.